I'm Erin DiCaprio, virologist and food safety specialist in the Department of Food Science and Technology at UC Davis. Here we'll discuss foodborne viruses as compared to SARS-CoV-2, the virus that causes COVID-19. Viruses are not considered to be living organisms. They are obligate intracellular parasites, meaning viruses require a specific type of host cell and a specific type of organism to be able to multiply or increase in number. This means they do not have the ability to replicate or increase in number in foods or in the environment. Viruses also have no type of energy generating mechanism, i.e. cellular respiration. Viruses are essentially rationally designed combinations of nucleic acids, proteins, and sometimes lipids that have the ability to modulate a host and that host cell's own cellular machinery in a way to make additional viral copies. There are two basic structures of viruses, non-enveloped and enveloped viruses. Non-enveloped viruses are the most simplistic of the two types of virus structures. Non-enveloped viruses are comprised of nucleic acid, which is coated or encapsulated in a protein. The viral protein that encases the nucleic acid is called the capsid. The viral capsid is formed from a protein encoded in the virus's genome, and the capsid may be comprised of multiple copies of one or several different viral proteins, depending on the virus type. The genetic material of viruses may be either DNA or RNA, depending on the viral type. Enveloped viruses are more complex in structure compared to non-enveloped viruses. On the exterior of the viral particle is a lipid bilayer that is derived from the host cell's own membranes. Within the lipid envelope is the viral nucleic acid, and this nucleic acid is encased in viral protein. Depending on the type of virus, there can be many other viral proteins associated with the nucleic acid or in the space between the nucleic acid and the envelope called the tegument. Another very important structure are viral proteins that are associated with or embedded in the viral envelope. These proteins include those important for viral cellular receptor attachment, viral membrane fusion, envelope stability, and subsequent budding or release of new viral particles from a host cell. To render a non-enveloped virus non-infectious, you can damage the capsid or the nucleic acid. An envelope virus can be inactivated by damaging the envelope proteins, by damaging the envelope itself, by damaging other viral proteins, or through damage to viral nucleic acid. Generally, envelope viruses are much easier to inactivate compared to non-envelope viruses because the viral envelope, the lipid bilayer, can be destabilized by many sanitizers, surfactants, through mild heating, and by drying out. They also have multiple targets that can render them non-infectious, including external proteins on the envelope. The capsid of non-envelope viruses is generally more heat resistant than the lipid bilayer of envelope viruses. Also, it has been shown that non-envelope viruses are very stable under dry conditions. Very harsh sanitizers must be used to inactivate non-envelope viruses. There are many types of microorganisms that cause foodborne illnesses. This slide lists them from the most resistant to the least stable. This means resistance to heat, sanitizers, and desiccation. Prions are pathogenic agents that are transmissible and induce abnormal folding of specific normal cellular proteins called prion proteins that are found primarily in the brain. The prion disease that likely everyone has heard of is bovine spongiform encephalopathy, BSE, also called mad cow's disease. Prions are the most stable foodborne pathogens and can only be inactivated by extremely high heat and harsh chemicals. Mycobacterium is a genus of bacteria that includes several causing serious disease in humans, Mycobacterium tuberculosis of note. These type of bacteria have a very different structure compared to other types of bacterial pathogens, and the structure of Mycobacterium make them very stable. Some types of foodborne bacterial pathogens have the ability to form spores that have high resistance in the environment. However, normal growing bacterial cells, i.e. vegetative bacteria, are not very resistant to disinfectants or heat. though many can survive in the environment for long periods of time if conditions are ideal. Non-envelope viruses, including norovirus, poliovirus, adenovirus, and others, are similar to bacterial spores and mycobacterium in their resistance to heat and sanitizer inactivation. Due to this fact, they remain infectious in the environment for long periods of time. Envelope viruses, including influenza viruses, coronaviruses, measles virus, and others, generally do not survive for long periods of time in the environment outside of the body. They can also be easily inactivated by heat and chemical treatments. Listed here are the major food and waterborne viruses grouped by the type of disease they cause in humans. All of the viruses listed here are non-enveloped and are transmitted by the fecal-oral route. This means you have to ingest these viruses to get sick. The primary site of replication for all of these viruses is the digestive tract. Many, such as hepatitis A virus, hepatitis E virus, and poliovirus, can also be subsequently replicating in other tissues after the virus has entered the bloodstream, leading to more serious disease outcomes. Since all these viruses are non-enveloped, they can remain infectious on surfaces, in food, and in water for weeks to months. There is currently no evidence of transmission of SARS-CoV-2, the virus that causes COVID-19, via food or food packaging. SARS-CoV-2 is an envelope virus, and data suggests it only remains infectious for a few days at most outside of the body. Also, the transmission mode of the virus is via respiratory droplet, meaning the virus must contact your mucous membranes either via aerosolized droplets created by someone coughing, sneezing, talking, etc., or by you touching a surface where these droplets have settled and then touching your own mucous membranes. These characteristics make it unlikely that food would be a vehicle for transmission. There is no such thing as zero risk, but at this point in time, I characterize the risk of food or food packaging being a vehicle for COVID-19 transmission to be very low. However, if a person with COVID-19 were to cough, sneeze, etc. on a food or food packaging, there could be infectious virus present on those surfaces. The best advice is to follow standard safe food handling recommendations. Wash your hands after handling food or packaging and before eating. Also, someone with COVID-19 should not handle or prepare foods. It is believed that cooking will inactivate the virus based on research conducted with SARS-CoV-1. Therefore, cooked food should not harbor infectious virus.